branch of Svetoslav Fedorov Eye Microsurgery Institution Eye Microsurgery. Congenital aniridia is a severe penunculaar defect of eye development, the effects of which can lead to blindness. Malformations in aniridia include changes in the anterior and the posterior segments of the eye. In addition, congenital aniridia is often accompanied by systemic manifestations of neurological and metabolic disorders. As a result, more and more researchers tend to call PEX6-associated aniridia as aniridic syndrome or PEX6 syndrome. It is a rare genetic disorder characterized by complete or partial absence of the iris, foveal hyperplasia, resulting in reduced visual function and development of nystagmus. The name of PEX6 syndrome is due to the fact that more than 95% of cases of isolated aniridia are associated with a mutation in the PEX6 gene. 30% of cases are de novo mutations, sporadic cases of aniridia. In other cases, the disease is familial. Aniridia is inherited in autosomal dominant mode with high penetrance and considerable phenotypic heterogeneity. In 85% of cases, it occurs as an isolated malformation, approximately in 13% as part of WAGR syndrome. Other syndromes account for approximately 2 to 5%. The PEX6 gene is a member of a large family of genes that regulate transcription of other genes and is involved in the early development of the eye. PEX6 is expressed in the forming eye, the brain, the olfactory bulb, neural tube, digestive tract, pancreas. This gene is located on the short arm of the chromosome 11 and refers to genes encoding protein regulators of embryonic development of the eye. Therefore, patients with congenital aniridia can have a variety of sensory defects, including smell and hearing deterioration. In addition to clinical evaluation of the disease, this malformation requires genetic confirmation for differential diagnosis with other diseases associated with congenital aniridia, such as Peters anomaly, Rieger syndrome, iridogodia dysgenesis, the WAGR syndrome, and others. The purpose of our study is a comprehensive assessment of the clinical picture of congenital aniridia in Russian patients observed in our clinic from 2003 to 2015. A complete ophthalmic examination of 68 patients from 51 unrelated families was carried out during the research. The average follow-up of patients was 3 years. Sporadic aniridia is recorded in 31% of cases. A family history of aniridia was noted in 41 persons, and in 6 cases, the nature of inheritance could not be traced. The mean age of patients at the time of treatment was 22 years. Aniridia was confirmed genetically in 51 patients. 33 different mutations in 37 unrelated families have been identified.